The alpha helix is an example of a secondary structure of proteins. The alpha helix is able to occur due to the hydrogen bonding of the backbone, so that partial negative and partial positive oxygen and nitrogen on that peptide bond are able to form hydrogen bonds on the backbone of the alpha helix, and we have our R groups pointing outwards. Now, when we talk about the stabilizing and disruptive forces that cause the alpha helix not to occur, we can talk about electrostatic interactions. Now we can imagine that if we have our alpha helix and the R groups are pointing outwards and all of the R groups have negative charges, we know that these negative charges are going to repel one another. And when they repel one another, this is going to disrupt that structure of the alpha helix, making it unlikely to occur. Now, one way we can get rid of these negative charges is if we protonate these R groups. And in order to protonate them, we would have to decrease our pH. So if we decrease the pH, we will make the alpha helix more likely to occur. Now, the second factor that could destabilize our alpha helix is too many bulky groups. So here I've drawn the alpha helix with phenylalanine and tyrosine, and we can see that if we have too many bulky groups, that's going to generate steric hindrance. It's going to be too crowdy, and the alpha helix isn't going to like the crowdiness, so the structure is going to be disrupted. And remember that when we have an alpha helix, we do not want too many bulky groups in general. We prefer amino acids with R groups that are not too big and not too small. Those are the ones we are looking for. So this is going to generate some steric hindrance, which is not beneficial to our alpha helix. Now, another factor that affects our alpha helix is proline. Now, remember that we talked about the hydrogen bonding of the backbone. And we know the hydrogen bonding occurs between the oxygen and the nitrogen, because nitrogen has a partial positive and oxygen has a partial negative, and these electrons are delocalized. But in order for hydrogen bonding to occur, we need hydrogen. Now remember that originally in our peptide bonds, we normally have a hydrogen pointing down. But since over here we have proline, and proline, the carbon of proline actually forms a bond with nitrogen, and nitrogen loses that bond with hydrogen. Now we no longer have hydrogen to take part in hydrogen bonding. So we will not get that backbone of the alpha helix, which is essential for stabilizing and creating an alpha helix. So we need that hydrogen bonding. But with proline, we lose that hydrogen bonding. Now, it's also important to note over here that the proline is actually going to restrict the alpha helix. So since it doesn't have that hydrogen, our alpha helix will not be able to make a turn properly. Now, another factor that can affect our alpha helix is the dipoles that we have at each end of our alpha helix. Now remember that one end is going to be our amino end, the other end is going to be our carboxyl end. Carboxyl end, we're going to have a partial negative, and the amino end, we're going to have a partial positive, because these two groups are creating a dipole due to one another. Now, if we have a partial positive, it would be better to have R groups that are negatively charged, because that'll create an attraction between the two. And over here, if we have a negative charge, partial negative, it would be better to have R groups that are that have positive charges. So for example, there's lysine or arginine. So if we have negatively charged R groups over here on that carboxyl terminus, like aspartate or glutamate, there's going to be some repulsion, because we'll have the negative charges of aspartate and glutamate at higher pH values, and that repulsion of the carboxyl terminus. So that's not favorable. Now over here, at our amino terminus, if we have glutamate and aspartate negatively charged, that is actually beneficial over here, because this is partial positive, and those two amino acids will have a negative charge charge at higher pH values. But over here, if we had R groups that have positive charges, like for example, a histidine at a pH lower than 6, or arginine or lysine, those positive charges are going to repel one another, and that is not beneficial. So the four characteristics in regards to stabilizing features and disruptive features of alpha helix is electrostatic repulsions generated at high pH values when we deprotonate our R groups and gain that negative charge, which generates that repulsion, which is not beneficial. R groups generate steric hindrance, which is not beneficial either because that generates crowdedness. We have proline, which causes nitrogen to lose that hydrogen, so we can no longer have that hydrogen bonding within our alpha helix that actually stabilizes the alpha helix. And the partial positive and the partial negative imposed on our alpha helix due to the carboxyl terminus and the amino terminus will affect our alpha helix. And that is the stabilizing features of alpha helix structures. And remember that the alpha helix is a secondary structure.